Evolution is the process that produces change in a population, in particular genetic change. The three forces acting in evolution are genetic drift, mutation, and selection. Genetic drift involves the sampling of genes from one generation to the next, and this sampling of genes leads to random fluctuations in gene frequencies in the population. Mutation is the ultimate source of genetic variation, and mutation comes about through changes in a gene. Selection is a systematic force that produces a favouring of one gene over another, and this also leads to changes in gene frequencies in a population.
Genetics started as the study of heredity. Today, we have identified how heredity works.
Heredity is the study of how information gets passed on from generation to generation through discrete units that we now call genes. And this was the missing piece in Darwin's account of how evolution works. Mendel described how heredity works in terms of discrete units. So genetic information is passed on from generation to generation. So there are two kinds of life form on this planet. One kind is bacteria, which have only a single chromosome. The other is eukaryotes. Eukaryotes are organisms like plants and animals, which have a double set of genes in each cell. In our case, we have 23 pairs of chromosomes. Each gene has two copies. One copy that comes from the mother and the other copy that comes from the father. So in studying evolution, it's useful to consider a species as a population or a collection of genes.
A gene is a unit of heredity that is coded on a section of DNA. Any gene exists not just in the organism, but because the organism exists in a population, through reproduction, the gene can be seen to travel in a pool.
When we study evolution, it's useful to see how the gene frequency changes over time.
All genes have variations. A good example is eye colour. While the eye does the same thing for everybody, we all have different eye colours. The differences arise from variations in the eye colour gene. And these variations exist in human populations at different frequencies in separate communities. To study the pattern of genetic drift, we concentrate on one gene's variations and its frequency within a population.
We're going to look at a demonstration of how genetic drift works. In this exercise, we'll be using these green and red counters. Each one of these represents one allele or form of the gene. And because the colour is the only thing distinguishing these two, it means that we'll be choosing these counters completely at random. Imagine there's a population of 6 individuals, and because each individual has 2 copies of the gene in question, there are 12 copies of the gene in the population. The other thing we assume in this is that there's random mating. This is not always true in nature, but it's a good starting point.
First, we blindfold the selector. This covers the concept of random. This is done to ensure the selector's taste will not influence the picks. Then, we prepare counters to be shaken in a jar. The jar contains our entire gene population of 12. The shake and the picking of the 2 counters covers the mating part of the random mating. The two counters represent the first creature of the next generation. Once we log the result of the two counters, we place them back into the jar. Then we shake and pick again, selecting the second creature of the next generation. We repeat this procedure until we have 6 creatures logged.
The other important assumption in this model is that the total population size is going to be kept constant. We're repeating this process over many generations in order to track what happens to the gene frequency.
After 6 lucky dips, we have our first generation. The ratio of these genes then defines the composition of genes for the next generation. In this generation, we found 5 red and 7 greens. So the next generation will consist of a pool of 5 red and 7 greens. By keeping track of how many genes there are in any generation, we can log the ratio of red genes and green genes from one generation to the next. This is called binomial sampling.
So the frequency of one color should change, fluctuating in this random manner from generation to generation.
What we find over time might look something like this. In the final generation, we can see that the red gene has gone extinct. We can repeat the whole process and find that green goes extinct, not red. It is totally random, but the procedure still possesses the trend that eventually one will win and the other will disappear from the gene pool.
So in this case, it took 13 generations for one to take over in the population. But it could have been 10, it could have been 18. It was a chance event that red went extinct. If we did this experiment again, of course, green can go extinct just as easily. And this is because we started the process with 6 greens and 6 reds. And in this case, there's a 50% chance that green wins. So it's important to remember that when the population size is small, genetic drift is very potent. The extreme case of this is called a bottleneck, and genetic drift operates very quickly through a population bottleneck.
A bottleneck happens when a small sample of a population breaks off from a larger group. The smaller sample taken may not have the same ratio of genes as the main group. This difference, together with a smaller population, combines to a great evolutionary effect through genetic drift. The effect of bottlenecks can be seen in the evolution of flora and fauna on isolated islands, such as the Galapagos.
So we performed this exercise just now using a population size of 6 individuals, which is very small. And in this case, fixation, which is replacement of the population with one gene, happened very quickly. If the population size is larger, drift operates more slowly, so it would have taken much longer to reach fixation if we had a population size of, say, 100 or 1,000.
We can repeat this procedure with a much larger gene pool, with, say, 100 instead of 12. What we find is that it might take a long time, but eventually we come to the point where one gene wins and the other becomes extinct. This, in essence, is how genetic drift works. Genetic drift pushes towards fixation at every generation. It is a constant, powerful, ongoing force in the evolutionary process. In different populations, the outcomes might be different. Two adjacent but isolated groups may turn in totally different results.
You get a lot of differences accumulating between populations simply because of these chance events. This is very important when we try to track populations because even though these genes are neutral, in that there are no differences between two forms, we can use them to locate individuals. So we might, for example, sample a bird from a population and ask the question of whether it's from population A or B. And because of these neutral differences between two populations, we can make this kind of assignment. So this is a useful genetic tool for us.
This is how populations isolated from each other for very long periods of time undergo what is known as speciation. Speciation is the formation of a new biological species.
Speciation occurs when one species splits off into two species, and this can happen through, for example, isolation. Then these differences accumulate through perhaps genetic drift, perhaps even selection. The important difference that might arise is biological reproductive isolation. So when members of these two populations are no longer able to breed with each other, then speciation has begun.
A good example of speciation can be found in the prosimian family of primates. The prosimian family of primates has two main groups. They are lorises and lemurs. Lorises live on the large Indian subcontinent, have large populations, and have only two species. Lemurs, on the other hand, live exclusively on the island of Madagascar. They have over 40 species on record. This is because Madagascar is not only geographically isolated, but also offers so many little isolated niches within. Given random mating and no particular selection, the ramifications of genetic drift are as follows. Genetic drift takes place faster in smaller populations. In larger populations, genetic drift is far slower. Genetic drift takes place faster in isolated populations. In non-isolated populations, genetic drift is much slower. Populations living right next to each other, but isolated from each other, may drift into being different species over a very long time through genetic drift. A mutation is a change in a DNA base or a chromosome. There are many types of mutation. Mutations are changes that occur in the DNA of some individual. An example of mutation is albinism. Albino animals lack the ability to produce pigments because of a mutation.
The transmission of genes from one generation to the next requires the replication of DNA. This is usually a very perfect process. Occasionally, though, there will be some sort of an error and the sequence that comes out will be slightly different. And that's essentially what mutation is.
All sorts of physical and chemical factors can bring about change in the DNA sequence.
There are many different kinds of mutation. The most important kind is perhaps point mutations. So just a single site along the DNA is changed from one letter to another. Other kinds of mutation include deletions, insertions, translocations. So this means that whole chunks of DNA are moved around and may be reversed. Mutations are very well studied in Drosophila. So this is a model organism of fly that's quite well studied. In that species, there are many examples of eye color change or wing shape change, this sort of thing. Another classic set of examples is antibiotic resistance in bacteria. Bacteria are very well studied in this regard, and we now know the mechanisms of resistance for many antibiotics.
A mutation can be neutral, deleterious, lethal, or advantageous.
If a mutation is deleterious, it will hinder the ability of the organism to survive and reproduce. Similar for lethal, except that in that case, the organism can't survive at all. If a mutation is neutral, this means that it has no effect on its ability to survive or reproduce. If a mutation is advantageous, that means that it confers a bonus for its survival, so its fitness is elevated.
The point about mutations is that in each organism there are countless mutations. The crucial thing to remember is that the number of neutral mutations far outnumbers the deleterious, lethal, or advantageous mutations in any single organism. In practical terms, what this means is that every entity, every organism is an experiment carried out by nature.
Mutation is the ultimate source of variation. Without mutation, you don't have variation. Without variation, you can't have evolution. So evolution needs mutation. Now, most mutations will end up being simply neutral or only slightly deleterious. And so it's only the occasional mutation that will lead to adaptive change. But that's enough to drive evolution in the long run. So in terms of these counters that we were talking about before, drift has the role of eliminating variation, which is the opposite of mutation, really. So if drift alone is operating, a population will end up being monomorphic, meaning all counters will be, say, green. In order for variation to be reintroduced, we need mutation. So you can imagine mutation as production of a single red counter in a population of green ones. And over the following generations, there is some chance that the red mutant will increase in frequency in the population. Most of the time it will simply go extinct, but because mutation is an ongoing process, you'll have these new red variants coming in, and occasionally one will enter the population reaching high frequencies.
Mutation is a mechanism by which alternative genes are introduced into the population. Mutation acts in opposition to genetic drift by increasing variation. Natural selection is the most famous of the main mechanisms for evolution, so much so that it is almost synonymous with evolution itself. Charles Darwin found varying finch species on the Galapagos Islands that convinced him that the environments on each island had impacted on the finch populations differently. Natural selection is an important force in evolution. It
It's through natural selection that adaptive changes occur in a population. And this is what Darwin was concerned with.
In nature, the environment acts to pick which organisms survive and which do not. The ones that survive continue to pass on their genes. Factors driving selection can be as simple as predation by stronger animals to more subtle things, such as impacts of weather and other events. The effect of nature may be very subtle to more drastic. Selection occurs when one trait is favoured over another by the environment. Over long periods of time, organisms change, adjusting to the challenges of their environment. This is known as adaptation. We can simulate the effects of selection on our previous model. Imagine instead of counters, Lego pieces.
In this case, instead of the two forms of a gene being identical, suppose that there's some subtle difference.
One Lego piece is 2 by 2, the other 2 by 3. Naturally, there is a greater likelihood of picking up the larger piece than the smaller piece. So once again, we run the simulation with 12 pieces, 6 apiece. The hand that reaches in is just a little more likely to pick up a larger piece than a smaller piece. Our results show that the advantage in the larger piece to be picked up leads to a gradual extinction of the smaller pieces. What is important to note is that even a slight selective advantage will eventually lead to significant changes in the composition of the population. Once again, this assumes random mating. Also, in a larger population, the same simulation will take a lot longer to reach the extinction point, but eventually it will. In a population of 100, the progress might look like this table. Clearly we can see the trend, but it is taking a lot longer for the trend to reach its logical conclusion. What this means is that in a larger population, the effects of natural selection might take a long time to manifest. If the selection criteria are more drastic, we will find that the process will be quicker. This can be shown in the demonstration where instead of 2 by 3, we have 2 by 4 pieces. With high selection acting in favour of the larger piece, we find that the small piece goes to extinction a lot quicker. How does selection interact with genetic drift?
When the population size is small, drift is powerful. This means that even if a gene is favoured by selection, drift may overpower the force of selection, and a mutant that arises which is advantageous may be driven to extinction by drift. Conversely, when the population is large, a selected variant of a gene will be able to sweep through the population unhindered by the effect of drift, because drift is weaker in a large population. A nice example of natural selection is fish. Extensive fishing in recent years has led to a decrease in the physical size of fish. And this is because when fish are netted, the smaller individuals are better able to escape, survive and reproduce. And as a result, the average size of fully grown fish is decreasing over time.
In this case, human fishing activity has become a natural selection process for the size of fish in the sea. Another example of natural selection around us is seen in hospitals. After years of using antibiotics to treat bacterial diseases, we now find that the bacteria that have survived the antibiotics have passed on the resistance into the gene pool. This has meant older antibiotics have become less effective than when they were first introduced.
It is sometimes possible to quantify the strength of selection. So, for example, suppose that we want to know how strongly antibiotic resistance is being selected in bacteria. Then we can compete sensitive strains against resistant strains and see the rate at which resistant strains replace sensitive strains in a culture. And that way we can actually try to quantify the strength of selection.
The process of selection is not always operating against our perceived human interests. Humans have been using selection in their favour for a long time, through breeding of crops and animals. This is known as artificial selection. Typically, we select for a trait we would like better fleece on sheep or sweeter fruit. We control the population size, and the smaller it is, the faster we reach our desired outcome. Selection is the environmentally driven mechanism of evolution that brings about change in a population. Populations adjust to the environment through adaptation. Even small differences in selective advantage can result in vast changes in a population. Through these simple models, we can see firsthand how genetic drift, mutation, and selection operate, giving rise to the dynamic phenomenon we understand as evolution.